Hello students, this is Mrs. Shweta Laddha from Fergusson College, Pune, and we are discussing about our first chapter, 12th standard biology, reproduction in lower and higher plants. Angiospermic plants, we discussed many topics. Last lecture, if you remember, we have studied the structure of male gamete, development of male gametophyte, structure of anatropous ovule, development of female gametophyte. We also discussed about double fertilization, pollen pistil interaction, what is syngamy, what is triple fusion, and what is the significance of double fertilization. Today I am coming towards the last part of this chapter where we will discuss about three important concepts, that is development of seed. After development of seed, we will discuss about development of embryo and apomixes, parthenocarpy, pseudocarpy, some different concepts related to seed. So let us begin with a PowerPoint presentation. So first we will see about the post-fertilization changes. You know that due to syngamy, first fertilization, when the first male gamete, it fertilizes the egg, it results in the formation of the embryo. And due to the second fertilization, the second male gamete, when it undergoes triple fusion, when it combines with the polar nuclei, the secondary nucleus, it results in the formation of endosperm. What happens and how this endosperm develops, we are going to discuss about this. So let us see endosperm formation. There are different types of endosperm formation. The first type is called as the nuclear type. Here, first division of the primary endosperm nucleus occurs, which is not accompanied. So look at this diagram. Here you can see a single primary endosperm nucleus, and how it is? It is triple N, and this undergoes division. You can see two nuclei form, many nuclei form, and so on. So here there is only nuclear division is taking place. It is not followed by cytoplasmic division. Many nuclei are produced freely, and at last cell wall formation is seen. Example, the coconut water in the tender coconut is cell wall formation never completed in this. Another example is maize. So again I repeat. So in this was the triple N primary endosperm nucleus. It divides to form two nuclei. 4, 8 and many nuclei, and many nuclei are formed, and at the end every nucleus it is surrounded by cytoplasm. So freely nuclei are dividing, cytoplasmic division takes place at the end. Such type of endosperm, when it is developed, it is called as nuclear type. Which example? The coconut water which we are drinking, it is having many nuclei. Cell wall formation is not completed in that, but even in maize such type of endosperm is developed. Now let us see the second type. In the second type, we will see about cellular type. Here there is cytokinesis after every karyokinesis. So this was our triple N, primary endosperm nucleus. It divided to form two nuclei, and if you can see, it is followed by cytoplasmic division and two cells are formed. Now this cell will again divide to form two nucleus, nuclear division followed by cytoplasm division. So every nuclear division is, or every karyokinesis is followed by cytokinesis. And so every nuclear division, that is karyokinesis, cytoplasmic division. Again nuclear division, karyokinesis, cytokinesis. So continuously it takes place. It is seen in Petunia, Datura, Balsam, Adoxa. You have to remember the examples, can be asked for MCQs. And so the endosperm, when it is developed, it is multicellular type of endosperm from the beginning. The white kernel in the coconut, आपण मराठी त्याला खोबरं म्हणतो, खोपरा. The white kernel part of the coconut, it is a cellular endosperm. Okay, so this is about the cellular type. Next we will discuss the third type, which is seen in Helobial type, Helobiaceae family. It is intermediate type. What does it mean? First division is accompanied by cytokinesis. So first this was our primary endosperm nucleus. It divided into two nucleus followed by cytokinesis. So two cell stages form. Of course one cell is big, one is small. The smaller one is called the chalazal chamber. The larger one is micropylar chamber. After this, The nuclear sac larger one and towards the chalazal end is smaller. Okay, I told you. So the one which is larger, it is micropylar chamber. The one which is smaller is towards the chalazal chamber. It occurs in asphodels. So first division is karyokinesis followed by cytokinesis. But after this, see what happens. In the larger chamber, continuous free nuclear division takes place. Okay, and many nuclei form. And at the end, what happens? The cytokinesis is preceded. So this is halobial type and it can be seen in asphodel. So three types we saw how endosperms are developed. One is the nuclear type, other is the cellular type, and the third is the halobial type. Okay. Now let us proceed. What happens to the endosperm? What is the fate of the endosperm? Endosperm, its function is to nourish the embryo. Nourishment of the embryo is its function. If it is completely consumed in the embryo formation, then the seed will not have endosperm. Such seeds where endosperm is completely consumed and there is no endosperm in the seed, it is called as non-endospermic seed. It is also called as X-albuminous seed or non-albuminous seed. Example, pea, beans, groundnut, all these dicots, they have cotyledons which are storing the food. They do not have endosperm because endosperm is completely used when the embryo was developing X-albuminous or non-endospermic seed. If endosperm persists, if it is still there after the formation of seed also, then we call such seeds as albuminous seeds or we call them as endospermic seeds. Example, castor, arundi, coconut, rice, wheat, maize, barley, sunflower. These are the example of albuminous or endospermic seeds. Now let us proceed with the development of the embryo and the seed. Now embryo development. We studied about endosperm development, three types of endosperm development and two types of seeds which are formed whether endosperm is present or it is not present. Correct? Now we will see how embryo development occurs. A wonderful process. It is almost similar in both monocot and dicot till half part and then it changes in monocot. Let us see. So the process of development of embryo is called as embryogenesis. After fertilization, first male gametes unites with the egg, syngamy occurs and now embryo will be formed. That process is called as embryogenesis. So you can see in the first picture what is there? Fertilized egg you can see. The zygote forms a wall around it and it is converted to form a oospore. So this zygote after fertilization, it forms a wall surrounding it and what do we call it now? Oospore. The oospore now divides transversely to form two cell stage. Look at this picture. You can see two cell stage. The upper cell is the apical cell. Lower cell you can see the basal cell. The oospore it divides transversely to form two cell stage. The larger basal cell is called as the suspensor cell. The basal cell, upper cell is small. Larger cell is at the bottom. This basal one, it is called as basal or suspensor cell towards the micropyle. And where is it present? It is present towards the micropyle inside the nucellus. You remember the ovulca structure, micropyle, and towards the micropyle this suspensor is present. And a small apical cell which is also called as the embryonal cell. So upper cell is called as the embryonal cell, lower cell is called as the suspensor cell. Where is the suspensor cell? Towards the micropyle. Where is the apical cell? It is towards the interior side of the embryo. Okay. Then what happens? Then it starts dividing continuously. The suspensor also starts growing and it will push the developing embryo inside the nucellus so that it will derive the nourishment, okay, from the endosperm. And let us see what happens. Initially this was the single cell, apical, basal. This basal cell continues to divide and it forms a suspensor cell. The basal cell is still there and it will push the developing embryo inside. Now, when this two cell stage continuously divide, look at the apical cell, what happens? It becomes 8 cell stage which is called as the octant. Then it becomes rounded in shape called as globular. Till octant stage, the development of embryo in both monocot and dicot is similar. Now it changes little bit in dicot. In dicot, then it becomes globular stage. Slowly it becomes triangular. What is becoming triangular? The developing embryo. What is the role of the suspensor? The role of the suspensor is just to push the developing embryo inside the nutritive tissue. Then slowly it becomes heart shape. Then it becomes horseshoe shape. Horseshoe shape is called as torpedo. And finally, a mature embryo which is developing, it consists of these two cotyledons, because we are seeing of dicot. Two cotyledons are formed, the radicle is formed, okay, the plumule will be developed and a complete embryo is developed. So form from this two cell stage, again I repeat, until the formation of organs, the embryo is commonly called as proembryo. So initially we will call it as proembryo. Basal cell divides transversely to form a row of cells which are called as the suspensor. You can see the suspensor. What is the function of suspensor? Suspensor is just pushing the developing embryo for getting maximum nourishment. And the lowermost cell of the suspensor, it is called as hypophysis. The suspensor pushes the developing embryo deeper into the endosperm for proper nourishment. It functions as a hostorium. Hostorium, one which will suck the food. Here it is helping the embryo to suck the food, to derive the food from the endosperm. Now, so the apical cell of the two-celled proembryo undergoes a transverse and two vertical divisions at right angle to form octant. So here it is explained how the two-celled, it becomes an octant. So how it is becoming an octant? Two vertical divisions at right angles to each other and one horizontal division, it will form octant. Then 8 cells of the octant proembryo undergo many divisions to form spherical mass, the globular stage. From the globular it becomes triangular, then heart-shaped, then torpedo. Torpedo is also called as horseshoe-shaped. Finally, it gets differentiated into embryonic axis. It has plumule, it has radicle, it has two cotyledon because we are talking about dicots, and only single cotyledon will be developing in monocot. So this is how embryo development occurs. I hope it is clear to you. Now, embryo development, I told you it is similar till octant stage. Now let us see about seed. Now, any seed, it consists of cotyledons, the embryo part I am talking, not about the endosperm. Embryo part, it consists of cotyledons and the embryonic axis. If there are two cotyledons, we call that seed as dicot. If single cotyledon, monocot. Embryonic axis consists of a point of attachment, hypocotyle, epicotyle. Epicotyle give rise to plumule, plumule will give rise to shoot. Hypocotyle will give rise to radicle, radicle will give rise to root. I'll just draw a brief rough diagram so that you understand. So these are our two cotyledon. This is the embryonic axis. On the top, I'll use green color. Embryonic axis give rise to epicotyle. Epi means above the cotyledon, and which will give rise to plumule. So this is the plumule. Plumule will give rise to the shoot. Let us use brown color to show the root. Below the cotyledon, below the embryonic axis, give rise to hypocotyle. Hypo means below, which will give rise to radicle, and radicle will give rise to the root. So this is radicle, this is hypocotyle, this is epicotyle, this is plumule and cotyledon. And this central region, it is called as embryonic axis. We'll see it again. So this is a seed. Seed is covered by seed coat. It has hilum. It has still the opening, which is called as micropyle. In the center, this is the embryonic axis. Upper part, epicotyle, giving rise to plumule. Lower part, hypocotyle, giving rise to radicle. After fertilization, wall of the ovary will form the fruit wall, which is called as the pericarp. You have studied the structure of pericarp last year in 11th standard. Okay, so pericarp is the fruit wall which consists of epi, meso and endocarp. Okay, fertilized ovules. After fertilization, the ovules will form the seed. We have already discussed seeds are of two types. Endospermic seeds called as albuminous seeds having endosperm. Example, castor, wheat, jawar, barley, sunflower. And non-endospermic seed without endosperm, also called as X-albuminous or non-albuminous. Example, pea and groundnut. Outer integument of the ovule forms testa of the seed. Very important. We studied ovule was bitegmic. So outer integument forms testa of the seed and inner integument it forms the tegmen of the seed. So what are the seed coat? The seed coat it consists of two covering, testa and tegmen. Micropyle in the seed. What is the function of this micropyle which is still there in the seed? It serves as a channel so that oxygen will pass through it, water will pass through it, and then it will help in germination. Look at this picture, cotyledon. Above the cotyledon, epicotyle you can see, and above the epicotyle you can see that plumule which is going to form the shoot. In this picture you can see below the cotyledon, hypocotyle, and below the hypocotyle Radicle will be formed. Radicle will give rise to the root system. So I hope you understood basic structure of an embryo, embryonal axis, cotyledons, hypocotyle, epicotyle. Okay. Now, monocot seed. Let us understand the structure of a single cotyledon seed, monocot seed. In monocot, the embryo, the single cotyledon occupies the terminal position and plumule is lateral. You can see here, first we'll see the upper part of, I'll use another color which will darken it. So this upper part, it is nothing but the endosperm. What is the function of endosperm? To provide nourishment to the developing embryo. This lower part, this part, it is nothing but the embryo, correct? And embryo will be nourished by the endosperm. So this is monocot endospermic seed. Entire monocot seed, it is covered with a seed coat. You can see a fruit wall and a starchy layer which is called as a alluron layer, which is protein, rich in proteins. Now the embryo, if you see, it consists of plumule, which will give rise to the root, shoot system, and the radicle, which will give rise to the root system. Radicle, it is protected by a layer which is called as coleorhiza, and plumule, it is protected by a layer which is coleoptile. How to remember these names? Little bit difficult, but always remember, students. Plumule, coleoptile, coleop. So there is coming a P in coleoptile. Radicle, coleorhiza. Rhiza is root, isn't it? So coleorhiza. So in monocot seed, I'll read out, the single shield-shaped cotyledon is called as the scutellum. The scutellum is a shield-shaped structure of the cotyledon. The protective sheath of the plumule is called as coleoptile. I have discussed this. And the protective sheath which is present in surrounding the radicle, it is called as coleorhiza. Finally, ovule is transformed into seed and ovary into the fruit. So I hope you understood the structure of monocot seed as well. Now seeds can remain dormant for a long time. It may happen that the seeds are not germinating for many years. Yeah, it is possible. One example given in our textbook is lupine, Lupinus articus. The seeds of lupine, Lupinus articus, they remain dormant for 10,000 years. Okay, and after that they germinated. So one more example, this is the seeds of lupine, Lupinus articus. Okay, and they remain dormant. Dormant, they did not germinate. Maybe favorable conditions are not there, water is not there, XII is very hard, it is not broken. Hundreds of reasons can be there why they did not germinate. There is another example called as Phoenix dactylifera. This Phoenix dactylifera, it is a date palm seed which is found at the palace of King Herodes. During the excavation was found to be dormant for 2000 years near the Dead Sea. So if you look at the picture, it looks like a khajur, kharik what we call. Yeah, it is a type of dactylifera, date palm only. A special type which is called as Phoenix dactylifera. And the seeds were found at the palace of King Herodes during excavation. And they had been dormant for 2000 years near the Dead Sea. Maybe the conditions were not suitable for them to germinate. So seeds, it is possible for them to remain dormant in the land without getting decomposed for many, many years due to some reasons. You remember these names? Lupine, Lupinus arcticus, Phoenix dactylifera. Lupine, Lupinus arcticus were dormant for 10,000 years, whereas Phoenix dactylifera were dormant for 2000 years. Pictures is not given in our textbook, but giving these pictures will make you easy to remember the names. Now some seeds are short-lived. We saw just now example of the seeds which are living dormant for thousands of years. But there are some seeds which live for very, very short time. Example, citrus seeds. All the seeds belonging to the citrus family, limbu, lemon. And some tiny seeds are easy for dispersal. Some seeds are so tiny. You can see in this picture, this seed is of orchid or striga or orobanca. In 0.5 millimeter, there are so many, 3 seeds are there. So they are very, very tiny. And if the seeds are very, very tiny, it will help their dispersal. If they are dispersed here and there, it will help in growth of the plants. So this is some peculiar characteristics about seeds. Now let us discuss about pseudocarpic fruits. Pseudo means false, carps means fruits. So let us see about false fruits or pseudocarpic fruits. Example, as you can see in the picture, apple, strawberry. The fruits which are formed from any part of the flower, but not ovary. Okay, the fruit which is formed from any other part of the flower, but not from the ovary. Such fruits are called as pseudocarps, or they are called as false fruits. Look at this picture. In this picture, you can see an apple. Actually, this is the pericarp. Okay, the ball of the ovary, and this the fruit, and this is the seed. So this part is the fruit, but do you eat that part? Rather, when you cut an apple, you will throw away that part, isn't it? What becomes edible? The thalamus becomes fleshy, edible, sweet. So such fruits where we are eating some other part, not the fertilized juicy ovary, that is a pseudocarp. In apple, in pear, the thalamus forms the fruit. The actual fruit we throw off, and thalamus becomes juicy and we eat the thalamus. Even in strawberry, thalamus is becoming edible. Strawberry, cashew, these are some examples. Pear, apple, pseudocarp fruits. So I hope you have understood the term of pseudocarpic fruits. Now let us understand the concept of Parthenocarpy. Parthenocarpy, example banana. This term was coined by Knoll in 1902, Parthenocarpy. Listen carefully. When fruits are developed without fertilization, they are called as parthenocarpic fruits. Students, you always get confused between pseudocarp and parthenocarp. Pseudocarp. How to remember? Pseudo word false. Carp is fruit. False fruit. We are not eating the fruit actually. We are eating some other part which has become fleshy. Thalamus in case of apple, pear, strawberry. Now parthenocarpy, we are eating fruit. The fruits are formed, but they are formed without fertilization. Such fruits are called as parthenocarpic fruits. It occurs naturally in some varieties of pineapple, banana, papaya. These varieties, they produce parthenocarpic fruits. In these plants, it seems that the placental tissue in the unfertilized ovary, they produces auxin, indole 3-acetic acid, and these hormones, phytohormones, are responsible for enlargement of the ovary and formation of fruit. These fruits are usually seedless because no fertilization and development of a fruit takes place. So these are parthenocarpic fruit which are developed without fertilization. Next, apomixes. Very important part. What is apomixes? It is a phenomenon of formation of embryos through asexual method of reproduction, that is without formation of gametes or without fertilization. When fruits were formed without fertilization, we call them as parthenocarpic fruits. Now we are saying that seeds are developing, embryos are developing without fertilization, without formation of gametes, then we call them as apomixes, and such seeds are called as apomictic seeds. It is unusual sexual reproduction where there is no meiosis, no syngamy, no fertilization, but fruits are developed or seeds are developed. Seeds are developed. Embryo develops in the ovule and ovule develops to form the seeds. In apomixes, if a gametophyte organ or a cell produces embryo-like structure without fertilization, it is called as apogamy. Apo, absent. Gamy, gametes. Absent of gametes. No gametes are formed. No gametophyte organs are formed. No gametes are formed. Still embryo is formed without fertilization. So it is apogamy. Similarly, if a gametophyte, when diploid sporophytic cell, the diploid sporophytic cell, if you might be remembering the nucellus, it is having this diploid sporophytic cell. They produces a diploid without undergoing meiosis into a seed, then we call it as apospory. Apospory, without developing meiotic spores. The diploid sporophytic cell only will produce into a, without undergoing into meiosis, it will develop into a embryo. Example, it occurs in oranges, in mango. Let us see apomixes. So apo, absent, mixes, mixing of gametes. No fertilization, still embryo is formed. No gametes are formed, still embryo is formed. No meiotic occurs, the sporophytic cell directly becomes into embryo. I hope you understood the concepts. There are three categories of apomixes: recurrent apomixes, non-recurrent apomixes, and adventive embryony. Repeat these words. Recurrent apomixes, non-recurrent apomixis, adventive embryony. I'll try to explain you all the three concepts slowly. First, we'll see recurrent apomixis. The embryo sac develops from, listen students carefully, diploid egg mother cell. Diploid egg mother cell. The egg mother cell is diploid and that only is developing into embryo from some from other diploid cells of the embryo sac. I hope you remember nucellus. The nucellus inside the ovule, it is having number of diploid cells, and those diploid cells of the embryo sac without fertilization, if they are developing into an embryo, then we call it as recurrent apomixis. Consequently, the egg has the normal diploid number. Here the egg is not haploid in this case, it is diploid. It has two sets of chromosome and therefore the chromosomes will be same as in the mother plant. This happens in Taraxacum. Okay, in Taraxacum you can see recurrent apomixis where diploid egg mother cell or some other diploid cell from the embryo sac without fertilization will develop into embryo. Let us see non-recurrent type of apomixis. The embryo develops here. It develops from the haploid egg cell. Haploid, it is not diploid, but that haploid egg cell, haploid parthenogenetically without fertilization, or some other haploid cell in the embryonic sac without fertilization will form into an embryo. So your embryo will be haploid. In recurrent apomixis, embryo is diploid, 2n, because it is developing from diploid egg. In non-recurrent apomixis, it is haploid because it is developing from haploid egg cell without fertilization parthenogenetically. Plants produced are haploid and they are sterile. As they are haploid, they are sterile, they cannot reproduce. Example, Nicotiana. And the third type is adventive embryony. Here there may be a mega gametophyte in the ovule, but the embryos arise from the cells of the nucellus. Everybody of you have known what is a nucellus. Nucellus, one of the cell of the nucellus, it formed embryo sac, isn't it? Here one of the nucellus, it forms directly into an embryo. And it is seen in mango, it is seen in orange, and it can be also seen in lemon. So three types of apomixis. Recurrent apomixis, in short, if you want to remember, diploid egg mother cell give rise to embryo. Non-recurrent apomixis, haploid egg cell give rise to a embryo. Adventive, one of the cell of the nucellus give rise to embryo. Here also it will be diploid in nature, here haploid in nature, and recurrent diploid in nature. So in adventive, it is also called as sporophytic apomixis or sporophytic budding or nucellar embryony because it is developing from the nucellus. Yes, students. So we have come towards the end of a very big chapter, very important chapter from the exam point of view. While revising this chapter, you start with simple definitions like what is pollination, what are the different types of pollination, what is siphonogamy. Then you start slowly studying about the parts of the flower. Okay, in parts of the flower, try to understand what is anther, the anther wall, the regions of the anther wall, how anther wall dehiscence takes place, how pollen grain is formed, what is the structure of pollen grain, how pollen grain develops into a male gametophyte. Then you come towards the ovule. Different types of ovule I have taught you. In that, discuss and revise about anatropous ovule. In anatropous ovule, then you study how the anatropous ovule has nucellus, how embryo sac is formed, and how female gametophyte is developed. Then you study about how fertilization takes place, double fertilization. Double fertilization, you should know the definition of what is syngamy, what is triple fusion, what is the result of syngamy, formation of embryo, what is the result of triple fusion, you should know formation of endosperm. Then we studied how development of endosperms takes place by three methods, the cellular type, the nuclear type, and the halobial type, and then how development of embryo. Finally, fruit formation, seed formation, and then some concepts which we have studied at the end like parthenocarpic fruit and pseudocarpic fruit. So I hope you understood the chapter. Thank you.